afternoon, everybody. Welcome back. Today we're going to learn how to build virus particles. I brought a couple with me today so that you can see them. Here's my favorite virus of all. This is polio virus. And the cool thing about this model is that inside it is the viral genome, so that you can see. This is actually a beaded genome. It's made with 4 different color beads. Each one is a different base, and it's in the right order. Somebody, a virologist, made this for me a couple of years ago. I'm not going to take the RNA out yet. We'll save that for next time when we talk about virus entry. But I wanted to show you this beautiful shell. We're going to talk about how to build this today. So that's polio. I also brought a model of HIV from giant microbes. You can buy these fuzzy things. And these are intended to get people interested in viruses, right? They're cute. They have eyes. This has a little red ribbon here. I'll tell you today why this is wrong. And then we have a cool one, which is made of little magnets, which we'll get to. And this is tobacco mosaic virus. And over the years, students have given me more and more magnets. This is half the size of the one I have in my office. All different color, but you can make a lovely helical virus out of it. So we'll talk about those today, virus structure. Now, the beautiful virus particles that we see here and in all the diagrams I show you have functions. I mean, the easiest thing you can think of is that these particles are meant to protect the genome, of course. They have to make a stable shell. So polio is a great example of that. This is a pure protein shell. Some viruses are made purely of protein as a shell to protect the nucleic acid. So the viral proteins that make up the shell have to assemble to form it. They also have to recognize the nucleic acid inside. We will talk about that later on in the course, because it's not good to make a shell if you can't incorporate specifically virus nucleic acids. You don't want to put cell nucleic acids in. And some virus particles have an envelope. Now, the HIV has a protein shell, but around it is an envelope, which you can't tell from this. And that envelope is derived from the host cell, of course. And there has to be a way for the virus to acquire that. So that's all built into the protein shell. In addition to protecting the genome, that's the major function of capsid proteins, to protect it. They may look beautiful. They're absolutely gorgeous. But they're thrown away once they deliver the nucleic acid. The nucleic acid is the main part of the virus, of course. And so the shell has to also give up the nucleic acid. It has to bind host receptors. We'll talk about that next time. It has to allow uncoding. It has to give up the genome. For viruses with membranes, the envelope has to at some point fuse with a cell membrane to deliver the genome. And then the genome has to go to the right place in the cell. It's not always just the cytoplasm. It could be early in the endocytic pathway or later. It could even be in the nucleus. And structural proteins play a role in that as well. Now, as we talk about structure of viruses, there are words I'm going to use. And again, I want to make sure you understand what I'm talking about. So here are a list of definitions. And we start with subunit. When I talk about the subunit of a virus particle, I'm talking about a single polypeptide chain. And that's diagrammed here. So here in the upper right is a schematic diagram of poliovirus, which is the virus there on the table. And this is made up of many copies of protein subunits. The protein subunits are the single polypeptide chains. Here they're labeled VP1, 2, and 3. VP is a common name for a viral protein. VP, viral protein. So that's a subunit. The structural unit is the unit from which capsids or nucleocapsids are built. Now this is a capsid here on the table. I call this the poliovirus capsid, and it's made up of structural units, which are made up of subunits in turn. So the structural unit of the poliovirus capsid is composed of 4 different polypeptides. Then we talk about capsids, which is from the Latin word meaning box. That's the capsid right there on the table. That's the protein shell surrounding the genome. Again, in the poliovirus image in the upper right, the capsid is shown there, and inside, of course, is the genome. Then we have another term which is often confusing from year to year called nucleocapsid. A nucleocapsid only exists in certain viruses when there are subassemblies. So poliovirus consists of a capsid and the genome, which is RNA. There's no nucleocapsid here. There's no subassembly. Now, if this had an envelope around it, which some viruses do, then the protein shell would be the nucleocapsid. And so for RNA viruses, which an example here is shown a vesicular stomatitis virus or rabies virus, the RNA protein complex inside the envelope is the nucleocapsid. It's a subassembly. And as we go through different viruses, I'll point out which ones have nucleocapsids, which don't. That's often, as I said, a source of confusion. And we have envelope, of course, which is a viral membrane in viruses that have it. Not all viruses, again, have an envelope, but some do. And those that do, it's derived from the host cell, of course. So here we have on this slide an example of a bullet-shaped rabies virus with an envelope. And then at the lower right is HIV, a retrovirus, where you have an envelope derived from the host cell. And within the envelope is a core composed of viral RNA and proteins. So that's a subassembly. That's a nucleocapsid. It's RNA protein. The term that I use, I've already used a lot in this course, virion. I've said virus particle and virion. When I say virion, it means the infectious virus particle. Because you remember, not all virus particles, as they're made in cells, are infectious. Some of them are broken. Some of them are mutated. So virus particle is the name for all of them. And virion, I specifically mean the infectious virus. I do anyway. Not everybody uses it that way, but I think that's the way it should be used. So let's again consider the size of virus particles in relation to other things that we might be familiar with. So a nanometer, we're going to talk about nanometers and angstroms a lot in this course. A nanometer, of course, is 10 to the minus 9 meters. 10 angstroms, that's 10 angstroms, 1 nanometer, 10 angstrom, or 0.001 microns. An alpha helix in a protein is about a nanometer in diameter, to put things into perspective. So here on the right is a protein alpha helix. DNA is 2 nanometers in diameter. Here's the double-stranded DNA alpha helix in this image on the right here. So it's about twice the diameter of a protein alpha helix. Ribosomes are 20 nanometers in diameter. Here in panel G is a ribosome. Poliovirus is 30 nanometers in diameter. So that virus particle sitting on the table there, that's polio. It's about 30 nanometers, slightly bigger than a ribosome. Most viruses are from 20 to, say, 250 or so nanometers in diameter, but there are some that are bigger, and lots of sizes in between. One of them, of course, is Pandora virus, shown on the lower part of this slide here. We talked a little bit about that the other day. These are 1,000 nanometers in length. They're not actually spherical, about 1,000 nanometers from one end to another. So much bigger than poliovirus, and of course has much bigger nucleic acid. So we go from nanometers all the way up to 1,000 nanometers when we talk about virus size. Now, a key point that you need to know and understand is that virus particles are metastable. Metastable. What that means is that they have to be stable to protect the genome as the virus goes from host to host, through the air or through fluids. It has to be very stable. It can't fall apart. But on the other hand, when the virus particle gets into a cell, it has to give up the genome. It has to come apart. So the particle has to be both stable and unstable. And that's why we call it metastable. So on this slide is illustrated the entry sequence for poliovirus, which we're going to talk about in more detail next time. But it illustrates this property. The virus particles, or the virions, outside of the cells are extremely stable. Poliovirus, the way it infects you is you swallow it. It goes through your stomach, it goes through your intestine, and there it can attach to cells. But of course your stomach is very acidic and full of enzymes. The intestines are alkaline with bile salts and other nasties, and the virus is totally stable to that. That's because it's a really nice icosahedral shell, as we'll see. Not all viruses can do that. Not all viruses are stable to extremes, but they are stable in the area of the organism that they infect and reproduce in. So virus particles are very stable. Well, yet when poliovirus gets into the cell, and here we're showing poliovirus binding to a receptor and being taken up by the endocytic pathway, at some point the RNA has to get out. So the beautifully stable shell has to destabilize. And we understand for a lot of viruses how that happens. It's quite interesting, and we'll talk about that next time. But here for polio, I'll just leave you with the fact that it's actually the receptor that binds the virus which catalyzes the RNA coming out of the virus particle. So for some viruses, the receptor does it. For other viruses, it's low pH or proteases in the cell. We'll talk about all those next time. So you have to have a trigger that unstabilizes or destabilizes the virus particle, because viruses floating around in the air or in cellular fluids, they don't want to just start uncoding, right? That would be totally non-productive. They want a signal, and the signal is typically provided by the host cell. Now, going into metastability a little more, so we understand, now you know on the surface what it means, but let's go at the molecular level what metastability means. It means that the virus particles have not attained their minimum free energy conformation. And that when they uncoat, that is what they do. They attain their minimum free energy conformation. But virus particles, as they're floating around in cells or in cell fluids or in the air, are quite stable. And in order to reach this, so that's shown on this graph by particle number 1, the red one. To uncoat or to release their genome, they have to get to this minimum free energy conformation, number 3, right, and the energy is on the y-axis, E. And to get there, they have to cross a high energy barrier. It's an unfavorable energy barrier. So in order to get over that, and that makes a lot of sense to have that higher energy barrier so that the viruses don't spontaneously get to that low energy situation and uncoat. There's some high energy barrier that requires energy to be put into the system in order to overcome. And that's shown by this intermediate 2 here. And where that energy comes from is it's put in the particle during assembly. When the virus particles are put together, energy goes into them, and we call them spring-loaded as a result. It's like a spring. The virus particle is like a spring, and if you have the right trigger to pop the spring, the virus particle will uncoat. The energy will be released, and the minimum free energy will be attained. So that energy put into the virus particle is used for disassembly, for the virus getting less stable. Again, when the cell provides the right signal. It can be a receptor, it can be some intracellular condition. That's the subject of our next discussion on Wednesday. But it has to be associated with the cell, because you don't want virus particles uncoding if there's no cell around to take up the genome and replicate it. So virus particles are metastable. Now, how can they be metastable? How could you design something that's incredibly stable at low pH and bile salts and high pH and so forth, even high temperatures for some viruses? How can you design that? Well, that's because virus particles are made to be very, very stable by taking many identical proteins and repeating them over and over to generate symmetry. And that gives you lots of contacts between the proteins, and that gives you a very stable structure. So created by symmetrical arrangement of many identical proteins to provide maximal contact. You don't build that virus particle using 20 different proteins. You take one or a few proteins and repeat them over and over, and that way you get maximal interactions between them, and you get really stable particles. So that's how you make a stable structure. An unstable structure results from that because those proteins are not ever, very rarely, covalently bound together. You should never say never in science, because there's always an exception. But in virology, most of the time, the virus proteins that make up the particles are rarely covalently bound. So that means they can come apart readily. And that's how they come apart upon infection. And upon the provision of a signal, they don't just spontaneously uncoat. On the provision of a signal, the proteins will come apart because of the energy built into them and release the genome. So that's the basis for metastability. And we'll look at this a little bit more next time when we talk about uncoding, and we'll also revisit it when we talk about how viruses are put together. So this concept of metastability is very important. So let's have a question about it. Virus particles, viral capsids are metastable because they must protect the viral genome outside of the cell. They must come apart and release the genome in a cell. They have not obtained a minimum free energy conformation. They're spring-loaded, or all of the above. So the answer is E. All of the above are correct, which most of you got. They have to protect the genome outside of the cell. That's when they're stable. They come apart to release the genome. They're unstable, so the two of them, metastability. They have not obtained a minimum free energy conformation. That's part of the definition, and they are spring-loaded. All of those are correct. We're going to talk a lot about the structures of viruses, obviously, here. And I want to tell you about the tools of structural biology that are used, and they're listed here. Electron microscopy, simply looking at particles for many years, from the 30s up until the 70s, gave us a lot of information. X-ray crystallography, first used for viruses in the 1970s, gave us detailed atomic level information about how viruses were made up. Cryo-electron microscopy, cryo-EM, we'll talk about that today, and X-ray crystallography, and nuclear magnetic resonance, or NMR. I'm going to talk about X-ray crystallography and cryo-EM mostly, a little bit about electron microscopy. If you're interested in learning more, Chapter 4 in the textbook deals with that. Now, the modern era of structural virology, when we begin to understand how viruses are built, starts in 1940 when Helmut Ruska, working in Germany, used an electron microscope to take the first pictures of virus particles. Here's the article in German, if you'd like to read it. It's online. You can actually see his microscope if you're in Munich in the, I think it's called the Technical Museum there. Anyway, this was a bacterium infected with bacteriophages. That's one of the first electron micrographs. And as I said, for the first time, we saw that they're particles, because we didn't know what they actually were. We thought they might be liquids. Now, electron microscopy works. You have to give contrast to the virus, because biological materials don't have a lot of inherent contrast, whether it be a virus or a cell. You usually have to do something to provide contrast. For light microscopy, we stain things to see them. For viruses, to do electron microscopy, we have to stain them as well. And we use electron-dense material. Remember, in an electron microscope, you're not putting light through the specimen, you're putting electrons. So to see a virus particle, you stain them with electron-dense materials that will repel the electrons, and you're seeing a negative image of the virus particle. And things like uranyl acetate, phosphotungstate, these are electron-translucent materials. And that we figured out in 1959. It has a very low resolution, 50 to 75 angstroms. If you think about the alpha helix, 10 angstrom in diameter, that's not a lot of resolution. You can see overall structures, but you can't see any details. And here on the bottom are some EMs, just to show you what's been done. Here on the lower left is adenovirus. This virus is an icosahedral capsid with little fibers sticking out at each vertice, as you can see here. And the next one is hepatitis B virus, which makes a mixture of different particles, both spherical and elongated particles. We have influenza virus, and on the right, poliovirus. You can see in this poliovirus prep that some of the particles are empty. Right? Most of them look closed, but a couple of them, 3 here, are empty. They probably don't have RNA, and then they're probably broken. They're not infectious. So that's one reason why not every particle is infectious. So you can see the size of viruses this way. You can see their shapes. You can see some details. You can see, obviously, the little fibers coming out of the adenovirus particle. But as I said, not a lot of information, although this allowed us to put viruses into different categories as a way of classifying them. Now, in the X-ray crystallography was developed long before it was used for viruses. The first virus structure determined by X-ray crystallography was in 1976. But protein structures had been determined long before that. What held up viruses was computational abilities. No one had a computer that could figure out the structure of a virus until the 70s. Before that, you could solve protein structures, globin, among others, the first proteins whose structures were solved. They used to make models by hand out of wood and metal, and they didn't need a computer to figure it out. But viruses they did, so that was delayed. What you have to do for X-ray crystallography, which can give us a resolution of between 2 and 3 angstroms. So you can really see everything. You can see the alpha carbon backbone. You can see the side chains as well. You have to be able to make crystals of the virus. So here is a poliovirus crystal. You have to purify the virus, tons of it, milligrams of it, and get it to form crystals. And that in itself is black magic. And it's very hard to do. Sometimes some things never crystallize, and sometimes they do. Sometimes they crystallize, and you can't get any information from them. So many people spent years doing this. what you do is you take your crystals, you bombard them with an X-ray source, and the idea here is that the X-rays will bounce off atoms in a repeatable way because of the crystal. You can record the bounces of the X-rays initially this was done on a film, on an X-ray film, and then the positions were measured. Nowadays they have scanners that can put this directly into a computer. But basically these diffraction patterns are generated by the virus in the individual atoms in the crystal. And from that you can get the structure of the individual polypeptides making up the virus particles. Now, more recently, people have adopted electron microscopy to solve the structure of virus particles. And here we call it cryo-electron microscopy, because it was figured out that if you freeze virus preparations in water, freeze them very quickly, it gives them contrast. So here on the upper panel is an example of one of these photographs. This is a purified preparation of a virus particle. It's frozen in water, and that gives it enough contrast that you can see it. Then what you do is you take photographs of individual particles. You take hundreds of them. The idea being that each particle is oriented slightly differently. And if you take enough pictures, you're going to get a three-dimensional image. It's kind of like a CAT scan. You go into a CAT scan and the X-ray spins around you and takes many X-rays, then they're computationally assembled. This is what we're doing here, cryo-electron microscopy. So you take individual particles, you actually then take their structures, you transform them by the Fourier equation, then you figure out ways to orient the centers of these particles, you merge them together, and then you do an inverse Fourier transformation to get the structure of the particle. So this is called a three-dimensional reconstruction, cryo-electron microscopy. Cryo because you freeze the particles. Now, cryo-electron tomography is a little different. Instead of taking pictures of so many different particles, you can actually tilt the stage on which the viruses are, kind of like tilting a person in a CAT scan and get the same information. Now, in 1985, the structure of poliovirus was solved at 2.9 angstroms resolution by X-ray crystallography. That's the structure on the left here. And it's drawn, of course, on a computer. You get a set of coordinates when you do three-dimensional structures, X, Y, Z coordinates for every atom. And you can use a graphics computer to make images and color them the way you want. Here in this structure of poliovirus, you can see where every atom is in 3 dimensions. Now, on the right is, just for comparison, a cryo-EM structure of poliovirus, which was done in a separate study. This is at a resolution of 20 angstroms. So you can see the surface of the particle, you can see surface features of it, but you can't see the details. There's just not enough resolution here in 20 angstroms. Now, cryo-EM is really much, much easier than X-ray crystallography because you don't have to make crystals. You just need a purified virus preparation and freezing it and collecting the data. And this can be done very, very quickly. And the resolution now has improved largely by improvements in the computer programs that are used to analyze the data. So here's an example of a study was published last year. This is Zika virus. The structure is done by cryo-EM. So Zika virus became a global issue mid-2015, end of 2015. This was published in March of 2016, 6 months, and 3.8 angstroms resolution. That's how fast you can do this now. So as soon as some new virus comes out that's really interesting, people jump on it. There are laboratories all over the world that can do this. This was published from the Rossman Laboratory at Purdue University, March 2016. Again, cryo-EM, 3.8 angstroms. You can see everything at that resolution. I made these 2 images myself. You can get free software to manipulate the data. On the left, I made a space-filling image. In this, I took every atom and made it a sphere and colored the different polypeptides differently, so you can see the symmetrical arrangement on the capsid. And on the right is an image where I just drew the alpha carbon trace, and there's some indications of beta sheets here as well. And of course, you get 2 different kinds of information. But I show you this just to emphasize how fast this can be now. You can do this within 6 months, and you can get very high resolution. So lots of labs are switching from X-ray crystallography to cryo-EM. This is the structure of one of those giant viruses, cafeteria Rowenbergensis virus. This is 300 nanometers in diameter, over 15,000 capsid proteins. This is done in the lab of Chun Xiao at the University of Texas, El Paso. He hasn't yet published this. It's taken him years to figure it out because it's so big. And he told me initially, when he would load all the atoms on his computer, it would crash routinely because it couldn't handle it. Now, of course, computing is much better. But he made this movie of it rotating just so that you can see how cool it is. And it's a very large virus. So these methods, this is done by cryo-EM, by the way, these methods can accommodate some of these largest viruses. So now you have an idea of what virus particles look like. Let's figure out how to build them, all right? And the key here is symmetry. Throughout most of the viruses we're going to talk about, there's symmetry involved. Not all of them, as you will see later on. And this whole area, understanding this, started with Watson and Crick, who you know, of course, as discovering the structure of DNA. By the way, they published their structure of DNA in 1953, and that's the same year I was born. That's why I'm a scientist, because I felt it in my blood. They also contributed to understanding virus structure, and they made a contribution which very few people know about. They figured out by looking at lots of different viruses, mainly by electron microscopy, that there are two kinds of symmetry used to build virus particles. The first is called helical symmetry. These are for rod-shaped viruses like tobacco mosaic virus. Helical symmetry is used. Or round viruses are built with platonic polyhedra symmetry. All that means, you know what the platonic polyhedra is solids like icosahedra. It turns out that icosahedra is the only platonic solid used to build round viruses. So either helical symmetry or icosahedral symmetry are used to build viruses. That was their contribution. And in both cases, identical protein subunits are used to build the virus particles. And here is an electron micrograph of tobacco mosaic virus. We've seen many times already. This is a rod-shaped virus built with helical symmetry. And on the right is poliovirus again, a spherical virus built with icosahedral symmetry. All right, so I want to explore these two kinds of symmetry now and explain to you how they work. Most of virus particles that we're going to talk about are built with symmetry rules, either helical or icosahedral. And these rules provide a way for viruses to self-assemble, essentially. And there are two rules that I want you to be familiar with. First, every subunit in the virus particle has identical binding contacts with its neighbors. Now, I have identical in quotes here because early on in this field, people thought the interactions were identical. And as you'll see later, they're not exactly identical. So rule one, each subunit has identical bonding contacts with its neighbors. In other words, repeated interactions of complementary surface leads to a symmetric arrangement. You take one protein and repeat it many times. You can build a particle from it, either a rod-shaped or a spherical particle, and that will give you symmetry. And the second rule is that the bondings are usually non-covalent, okay? And that means they're reversible so that the nucleic acid can get out. Also, in the process of assembly, if a mistake occurs in the assembly, it can be reversed. So you can make error-free assembly. It's kind of a quality control process for making a right virus particle. So two rules, identical bonding contacts of the subunits and non-covalent interaction. These rules allow you to make self-assembling virus particles, whether it's an infected cell or in the production of vaccines. We have two vaccines that we'll talk about later, hepatitis B virus and human papillomavirus vaccines. These are licensed human vaccines. These are produced simply by making the viral capsid protein in cells, in insect cells or yeast, and they self-assemble. Because of those rules of assembly, those two rules, the proteins, when made, fold properly, they assemble, and they make empty capsids. We call these virus-like particles, VLPs. And there's an electron micrograph of the human papillomavirus vaccine, which is an empty particle vaccine, no genome. You can just make the protein, and it assembles into a virus particle. You have all the information you need to make the nice shell without the nucleic acid. And For some vaccines, that's kind of a nice way to make a vaccine. It's quick, and you don't have to worry about infectivity. We'll talk about that much later when we talk about vaccines. So the rules of symmetry allow you to make virus-like particle vaccines. All right, let's look at them in some detail. Here is helical symmetry. Here we have a rod-shaped virus like tobacco mosaic virus, and typically they are made of a single protein that is bound to the RNA. So on the lower left is our single protein subunit, and these interact with each other to form a helix, and of course they also interact with the RNA. So you have protein-protein and protein-RNA interactions. So on the upper right is a schematic of this single protein interacting many times with each other and with the RNA. On the upper left is the actual cryo-EM structure of this capsid. All right, so we call this helical because the proteins together with the RNA form this helix structure. And you can see, again, the EM of tobacco mosaic virus, these are rod-shaped. The rods are nothing more than helices made up by these repeating interactions. These are buckyballs, which you can buy. They're very strong magnets. And if you imagine each ball is the protein subunit of a helical capsid, this is exactly how it works. And the magnets, of course, will stick to each other and make a nice alpha helix. This is exactly tobacco mosaic virus, except, of course, there's no RNA here. And otherwise, it illustrates perfectly. You can assemble and disassemble these particles. So if you find these are pretty cool, and you can make your own virus with them. All right, so that was tobacco mosaic virus. There are actually animal viruses that have helical symmetry. But as far as I know, all of them have envelopes around the RNA protein complex. So this one is a paramyxovirus, Sendai virus. It's related to measles virus. It happens to be a mouse virus. It has a nucleocapsid composed of RNA, a single capsid protein bound to RNA and wrapped up in a helical structure like the ones we've been talking about. But it's not naked like tobacco mosaic viruses. It's in an envelope. And the EM at the bottom shows you that. You can see the nucleocapsid spilling. This virus particle has broken, and the nucleocapsids are spilling out. Normally, there would be a membrane encasing the nucleocapsid, and they would stay in there until the virus infected a cell. All right, so the animal viruses that have helical symmetry, this structure is in an envelope. For some reason, the plant viruses can exist without having a membrane around them. Tobacco mosaic virus, here, this magnet model is tobacco mosaic virus. This is not technically a nucleocapsid because there's no substructure. This is just a capsid. Now, when you put a membrane around it, as you do for these paramyxoviruses, then the magnet or the RNA protein substructure becomes a nucleocapsid. That's the way you differentiate the two. So for Sendai, the RNA protein is a nucleocapsid because it's within a membrane. For tobacco mosaic virus, it's not a nucleocapsid. Here's just another example of an animal virus with a nucleocapsid that's helical. It's vesicular stomatitis virus. Now, this is a bullet-shaped virus, just like rabies virus, and many people who work on these tend to work on VSV, as it's called, because this doesn't cause serious disease in people, whereas rabies does, of course. Bullet-shaped virus, here's the schematic at the top. And the structures show you the EM on the left of the three parts of the virus. There's a tip, which has a closed end, which gives it the bullet-shaped appearance. There's a central portion, which is cylindrical. Then there's a base that's closed off. And this is nothing more than a helically built virus. In fact, there's one protein called the N protein, and it binds RNA repeatedly. Here's a structure of a number of N subunits with a piece of RNA, which you can't see here, but it's in green. And this whole structure is simply made up of N protein RNA complexes wrapped up in a helical structure. So the nRNA is shown in this greenish yellow color. There's another layer of protein on top of it, and then there's a membrane around it as well. So another example of a nucleocapsid RNA protein which has helical symmetry and is wrapped in a membrane. There are lots of viruses that look like this and that are built with this kind of symmetry. We've talked a little bit about the paramyxoviruses, measles, mumps virus, Sendai virus, which I showed you a picture of. Again, a helical nucleocapsid, RNA plus protein, wrapped in a membrane. Rhabdoviruses, rabies virus, vesicular stomatitis virus, we just looked at that. The influenza viruses are enveloped viruses. The genome is a nucleocapsid, a helically built nucleocapsid. It happens to be in pieces for influenza virus, but it's nothing more than RNA wrapped up in protein. And Ebola virus, the same thing. It happens to be filamentous, but inside this envelope, the membrane, is an RNA protein complex, a nucleocapsid built with helical symmetry. Okay, so that's how helical symmetry is used to make animal viruses with membranes. Next question, which of the following describes virus symmetry and self-assembly? The bonding contacts of subunits are usually covalent. The bonding contacts of subunits are usually non-covalent. Each subunit has different bonding contacts with its neighbors. Self-assembly of virus particles does not occur, or none of the above. The right answer here is B. The bonding contacts of subunits are usually non-covalent. And everyone did pretty well. No one picked C, D, or E, which is good. A few people picked A. The bonding contacts are usually covalent. They're not. If they were covalent, the particles would never come apart again. So think of it that way. In order for the particle to be metastable, the bonds have to be non-covalent. We talked about making rod-shaped viruses like tobacco mosaic virus, enveloped animal virus with helical symmetry. How do you make a round capsid now? Let's talk about that. So what do I mean by a virus with a round capsid like poliovirus here? This is obviously round or spherical. How could you do this with proteins that aren't round? Right? They have irregular shapes. And so here is an illustration of that problem. On the right is an electron micrograph of poliovirus, spherical virus particles, but the proteins that make them up are not spherical at all. So how do we do this? 2 clues. Clue 1, all round capsid have precise numbers of proteins. They're usually multiples of 60. They're not just random, any number at all. They're multiples of 60, like 60, 180, 240, 960. So that was one thing that clued them in. 2, spherical viruses come in many sizes, but the capsid proteins tend to be between 20 and 60,000 Daltons in size. So that was 2 clues that people got from working on virus structures for years and years, and that made them realize how you could build a round virus particle. These 2 individuals, Caspar and Klug, 1962, they published the solution. These were structural virologists. They knew from Watson and Crick that round capsids are icosahedrons. Watson and Crick had said platonic solids were used, and in fact, icosahedrons were the only platonic solids used to build virus particles. And secondly, subunits tend to be arranged in viruses as hexamers and pentamers. Now, why that is will become obvious in a couple of minutes. And what these 2 individuals proposed, which is now quite clearly correct, is that round viruses are built using the principles of icosahedral symmetry. So let's talk about icosahedral symmetry. What is an icosahedron? It's a solid, a platonic solid that has 20 faces. Each face is made up of an equilateral triangle, and that makes a closed shell with the smallest numbers of protein subunits. You could make an icosahedron with 1 protein repeated 60 times. The same protein, 60 copies, will make this lovely icosahedral shell. Now, this virus particle on the table here, poliovirus, is built with icosahedral symmetry. It doesn't look like an icosahedron. It's spherical, right? So these viruses are not actually icosahedrons. They're built with icosahedral symmetry, which means a solid with 20 faces, each triangle, and you can make the smallest one with 60 subunits. So this is correct. This doesn't look like an icosahedron. However, our HIV, they have made look a little bit like an icosahedron, which isn't correct. What's even worse is that this virus has a membrane. It's not an icosahedral virus. So I don't know where they got this from. We'll get back to that later. Now, a couple more things with icosahedron. When you take subunits and repeat them many times, you get axes of symmetry. So 5-fold axis, for example, All that means is that there are 5 copies around it. There's also a 2-fold axis, 2 copies, and a 3-fold axis. All right, and you can see each of those in these diagrams as well. We use these for reference points when we talk about viruses. Oh, at the 5-fold axis, there's an amino acid that's involved in receptor binding. So that's why you should understand these. So let's start with a simple icosahedral capsid. This is called a T=1 capsid. What that means will be evident in a moment. And here we can take one protein and repeat it 60 times. That's the simplest icosahedral capsid we can make. And in this virus, which we've diagrammed here as a spherical particle, which is correct, we've got the protein subunits shown as commas, just so that you can identify them. And these commas are all made of a single protein. And in fact, the protein for this virus is the structural unit. It's the same thing. For some viruses, the structural unit will be a collection of multiple proteins. But for this, it's the same thing. But what's important for this is that in these simple capsids, all the molecules interact with each other in the same way. Okay? They have head-to-head and tail-to-tail interactions. It's another reason why we use commas here, so that you can distinguish the head from the tail of the comma and see how all these proteins are interacting in the same way. So if you remember, the original idea about symmetry in virus particles would be that all the proteins interact identically, and that allows them to self-assemble. So these simple capsids fulfill that prediction. Interactions of all molecules with their neighbors are identical. All right, so that's a small virus, and we do have examples of these. One of them is a virus called adeno-associated virus 2, which is a member of the parvovirus family. Now this virus, you're going to hear about again multiple times in this course. It turns out it's one of the biggest viruses being used for gene therapy nowadays. It has a lot of properties that make it very good for if you're lacking a gene, to give you that gene back using an adeno-associated virus. So we'll talk about these quite a bit. Again, these are simple virus particles, 25 nanometers in diameter. They're made of 60 copies of a single capsid protein. Now, this is an illustration of the capsid on the upper right. The genome is a single strand of DNA, linear, with structures at the ends. We'll get into that later on, just so that you start to be familiar with it. Here's the single capsid protein on the lower left. And again, there's 60 copies of it that make up the final virus capsid. And again, they're arranged in icosahedral symmetry. And I think you can see here in the middle, well, first of all, the triangle illustrates one of the triangular faces of the icosahedron. And there in the middle is one 5-fold axis of symmetry. You can count 1, 2, 3, 4, 5 copies of that protein structure right around it. Right, so that's a simple virus. How do you make bigger ones? You don't use bigger virus proteins, because they're all about the same size, between 20 and 60 kilodaltons. You add more virus proteins. So that's how you make a bigger capsid, by adding more proteins, not by making bigger proteins. So here's an example of a larger virus particle. Here we have 180 identical protein subunits. So we start with 60 for the smaller viruses. You want to make a slightly bigger virus, you could add 180, which is a multiple of 60, following in the icosahedral rules. When you do this, there are a number of important changes. 2 of them are really important. First of all, you now have pentamers and hexamers on the surface of this particle. All right, now what do I mean by that? Let's find a pentamer here. Here's one part where you have 1, 2, 3, 4, 5 copies of the protein surrounding that 5-fold axis. That is a pentamer. But you can see that there are other parts of the capsid where you have 6. One here around this orange triangle, 1, 2, 3, 4, 5, 6. Those are hexamers. So that's what I mean by pentamers and hexamers. So by inserting additional subunits into the simple capsid, you now require the presence of both pentamers and hexamers. The only way you could do that is by making these 6-fold related areas. Now, as a second consequence of adding more subunits, we have different modes of subunit packing. In fact, they're labeled here orange, yellow, and purple. The orange, the yellow, and the purple commas, which are each proteins, all interact slightly differently. Now, you can't tell by looking at this picture, because you all have head-to-head and tail-to-tail interactions. And so they're not exactly identical, but they're similar. And that's the only way that you can build these bigger viruses. We call this quasi-equivalent. The bonding interactions are quasi-equivalent, but they all engage tail-to-tail and head-to-head. That's why I had identical in quotes on that slide a number of slides ago, because we used to think that the interactions between subunits were all identical until we started studying bigger and bigger viruses. And the only way you can make a bigger virus is to add more subunits, and that makes the interactions slightly different. But overall, they look the same, head-to-head and tail-to-tail, but the bonding environments are somewhat different. And the fact that you have pentamers and hexamers on the same particle should tell you that they have to be slightly different. In order to get those environments, you need differences. So that's a slightly bigger virus, and it's built, as are all the viruses that are even bigger, with quasi-equivalent. When a capsid contains more than 60 subunits, each occupies a quasi-equivalent position. And I've already said this to you, but the binding properties of subunits in different environments are similar but not identical. That's what we mean by quasi-equivalent. They're head-to-head, tail-to-tail, but there are subtle differences that allow for the construction of bigger virus particles. So here's an example of a virus built in this manner. This is SV40, a polyoma virus that we'll talk about quite a bit later on. It's about a 50 nanometer particle. It's made up of 72 pentamers of a single protein called VP1. And that gives a total of 360 subunits. So VP1 is a single viral capsid protein. First, it assembles into pentamers. So if you look at this, this reconstruction of the virus here, this is from the coordinates of the virus particle. Each of these knobby-like protrusions is a pentamer, 5 copies of VP1. And you can see in some of them they've been colored, 1, 2, 3, 4, 5. The gray ones you can't really tell. 5 copies of VP1 forming pentamers. And because this virus has 360 subunits, you know right away that there have to be pentamers and hexamers. That's the rule. And you can see here is a VP1 pentamer right in the middle, that purple one. It has 5 neighbors, 1, 2, 3, 4, 5. And then here is a VP1 pentamer with 6 neighbors, 1, 2, 3, 4, 5, 6. So they're mixed, pentamers, hexamers. This is quasi-equivalent interactions. We're building a slightly bigger particle than the one we looked at before, but the same rules apply. Quasi-equivalents, pentamers, and hexamers. By the way, these viruses have circular double-stranded DNA genomes. Now, on all these slides, you've seen this number, T number. That stands for triangulation number. It has to do with the way the icosahedron is constructed. The T number is the number of facets per triangular face of an icosahedron. So here is one of them here. There are 2 faces, 2 triangular faces next to each other, just a part of an icosahedron. So that's a T equals 1 virus. There is 1 facet to that triangular face. You don't see any divisions. It's another way to look at it. A T equals 4 virus is shown on the right. The equilateral triangle is outlined by the green pentamers, pentagons, and you see 1, 2, 3, 4 facets in that triangular face. So that's why it's T equals 4. Now, in reality, the calculation of the number involves a mathematical formula that is beyond us. We don't need to know about it. This is a simple way of explaining what it means. It simply means the number of facets in each triangular face of the icosahedron. And any capsid that has T greater than 1 is going to have both pentamers and hexamers, because we're making a bigger particle. T equals 1 is the smallest particle made of 60 subunits, 1 protein repeated 60 times. As soon as you make bigger ones, if you increase the T number, you're going to have hexamers in addition to pentamers. All right, so that's the T number. Now, why do I even tell you about it? Because when you increase capsid size, the T number also goes up, and this is a nice way to illustrate it. So here are 4 virus particles of increasing size, and we start at the top with a T equals 1 capsid. 1 protein repeated 60 times. We have the structural unit on the left, and the organization at the 5-fold axis of symmetry. So we have 1, 2, 3, 4, 5 structural units around the 5-fold axis. And here, that structural unit is basically the triangular face of the icosahedron. So there are no facets to the icosahedron. This is a T=3 virus, the next one in this series. If you look at the triangular face, there are now 3 facets. 1, 2, 3. We've added proteins. We've gone from 60 to 180 proteins. And by doing so, we've increased the T number from T=1 to T=3. By the way, if you have the T number, you can know the total number of subunits in a particle by simply multiplying it by 60. You want to make a bigger virus, T=4. Take a look at the triangular face of the icosahedron. You can see 1, 2, 3, 4 facets. That's shown in isolation at the left. You assemble those, you're going to make a bigger particle. Again, because you've got more proteins in that triangular face. And here's a T=13. The triangular face is divided up into 13 facets. I won't count them for you, but you can see they're all there. And again, as you add more and more proteins, you're increasing the number of facets. You increase the T number, the particle gets bigger. And this is the number of different subunits in the particle. So that's what T number is. I think that's the simplest way to look at it. Just look at that triangular face, and the number of facets in it is the T number. Which of the following are characteristics of icosahedral symmetry in viral capsids? Produces a solid with 20 faces, each with an equilateral triangle. Allows formation of a closed shell with 60 identical subunits. 5-fold, 3-fold, and 2-fold axes of symmetry. The T number describes the number of facets per icosahedral face, all of the above. The answer is E, which most of you got, all the above. All these things are characteristics of icosahedral symmetry. A solid with 20 faces. By the way, there are 12 5-fold axes in an icosahedron. 20 faces, 12 5-fold axes. Closed shell with 60 identical subunits. 5, 3, and 2-fold axes. And the T number is the number of facets per face. Now, on this model of poliovirus, this is a 5-fold axis I'm pointing to. There are 5 copies in blue around it. And the icosahedral triangle is shown here. But you wouldn't be able to tell me the T number unless I gave you more information. So I'd never show you a model of a virus and say, what's the T number? I'd have to ask how many subunits, for example. That might help you figure that out. But I want you to understand the concept of the T number being related to the size of the particle. Now, at some point, we make even bigger capsids with different proteins. And so it's not just taking 1 or 3 or 6 or whatever number of different proteins and repeating them over and over. Sometimes there are specialized proteins that go into making virus particles that don't fit in the scheme of symmetry. So here's an example of this. I'm going to go through just a couple of these more complicated capsids. This is adenovirus. Distinct components with different symmetries. So adenovirus is is built with an icosahedral shell, 150 nanometers. It's a T=25. So that means 25 subunit facets per icosahedral face. And with fibers at the 12 vertices. At the 12 5-fold axes of symmetry, there are fibers attached, and you can see those in the electron micrograph. So obviously the fiber conforms to the underlying symmetry, right? There's one at each 5-fold axis of symmetry. But in addition to the proteins that make up the capsid, 720 copies of one and 60 of another, there are other proteins interspersed throughout it that have a variety of roles. There are proteins bound to the DNA in the interior of the capsid. There are some other proteins interspersed throughout the capsid which are thought to help glue it together. They're not used to build the shell, but they're used to hold it together. So as viruses get more and more complicated, other proteins are thrown in for specified purposes. And as we talk about replication, we're going to address some of these as well. Here's another example of a more complex virus, which is very interesting. This is reoviruses. These are T=13 viruses, but that's the outer shell. These viruses are made of 2 concentric shells. There's an outer shell, which is made up of trimers of VP7, one of the capsid proteins. It's got a T=13 icosahedral symmetry. And you can see that on the diagram at the left. Those are the darker colored proteins. But then that surrounds a second icosahedral shell on the interior, which is colored in a lighter color. And that's a T=2 capsid made up of monomers of a different capsid protein, VP3. So basically 2 shells, one around the other. And they have different T numbers and slightly different symmetries. It's very interesting how they could interact with each other. Now, why do we have this? This happens to be viruses with double-stranded RNAs in them. Those RNAs remain in the capsid together with the RNA polymerase, and messenger RNAs are produced and sent out of the capsid. And when these infect cells, as we'll see next time, the outer layer is stripped off, but the inner capsid remains so that material can get in and mRNAs can get out. So it's a specialized requirement for this entire family of viruses. The bacteriophages, some of them have a very interesting appearance. So here are the tail, the so-called tailed bacteriophages. These are the ones everyone thinks of when you say virus to a fourth grader. They think of this because this is what they're taught. Of course, not all phages even look like this, but this is a lovely, unique appearance. And it's built with the 2 kinds of symmetries we've already talked about. So it has an icosahedral head. You can look at it and realize that it's an icosahedron, slightly elongated, but still an icosahedron. You can see the 5-fold axes of symmetry. And of course, within that is the DNA of this particular virus. Here's the cryo-EM structure of the head on the right here. 5-fold axes are colored pink. But then this virus also has a tail. The tail is attached to the head by a connector. The tail itself is nothing more than a helical structure, very much like tobacco mosaic virus, except here it's attached to a head by a connector, and the connector is not a kind of symmetry that we know about. At the very base of the tail is a base plate, and it has interesting tail fibers attached to it. The base plate is shown on the upper right here, and when these phages attach to a cell, the DNA comes out of the head, goes through the tail, and out the base plate into the host cell. So this tail is basically made up of helical symmetry for the tail and a number of other places, and icosahedral symmetry for the head. I want to show you the structure of part of the base plate, which is really interesting. This was just solved a couple of years ago. So some phages, the tail actually contracts and plunges a sharp spike into the host cell to make a hole through which the DNA comes out. And you can see that on the upper left picture here, the tail contracting and the spike exposed. On the lower panel B, shown in red, this is the actual structure of the base plate and this tail spike, as it's called. And this tail spike structure was recently solved, and that's shown enlarged in panels C and D, and it looks just like a spike. These are 3 copies of a single protein. It's a trimer. You can see red, blue, and green, and they taper from a wide area at the top all the way down to a spike. Why does it taper? Because that's the way the proteins are arranged. If you look at just the alpha carbon trace on the right, you can see the upper part of the spike is made up of a series of beta strands forming a nice beta sheet, and they taper to alpha helices at the bottom, or just loops, and they're held together by an iron molecule, which is shown in orange, that interacts with a specific residue of each of these 3 subunits. So it actually makes a spike. This is what pierces the cell membrane. Now, the spike probably has to fall off. We don't actually know what happens, because to allow the DNA to come through, it probably has to fall away. We don't exactly know how that happens. But what I like about this is beautiful form according to the function of this tail spike. Another interesting example of a more complex virus I want to show you is the Herpes simplex virus capsid. And I want to illustrate holes for entry and exit of DNA. So Herpes simplex virus is a bigger virus than we've talked about, 200 nanometers. So again, compared to polio, 30 nanometers. It's composed of a capsid, a larger capsid, but a similar icosahedrally built capsid. There's double-stranded DNA inside of it. But then the capsid, which is shown in part A here, is surrounded by a membrane. When these viruses infect cells, the membrane fuses with the plasma membrane of the cell. Then the capsid is free in the cytoplasm. And so the capsid, which is shown on the upper right, that's the cryo-EM structure of the capsid. It looks like just any other icosahedral capsid. You can see the 5-fold axes in red. So this is a big capsid, T equals 16. In addition to pentamers, do you think there are hexamers in it? Yeah, of course, anything bigger than t equals 1. Got to have hexamers and pentamers. So you can find them in there. But what's cool is that one of the five-fold axes is actually a portal, shown right here in panel D. It's a little hole. It's been modified. Just one of them is modified so DNA can come in when the virus is made and can come out when the virus is infecting a cell. This is actually an EM of the capsid showing you the portal here. And that's the cryo-EM structure of the portal itself. It's a lovely molecule. It's got motors built into it that help pull the DNA in or out. So what happens is the capsid of the virus floats down to the nucleus, it docks onto a nuclear pore, and the DNA comes out of the portal through the nuclear pore into the nucleus. Just beautiful. You see why I like viruses so much. They're just beautiful and incredibly functional. One year someone told me I was morbid for liking viruses because they kill so many people. And that's not the part I like, obviously. I think killing is an accident, but we'll get back to that. All right, so we talk about a lot of viruses that are naked icosahedron. Many of them have membranes, as we've just seen for herpes viruses. It's the lipid bilayer derived from the host cell. It's acquired by budding. We'll talk about this during assembly, but it's budding through a cellular membrane. It can be nuclear membrane, ER, Golgi. It can be the plasma membrane. It's always virus specific, so if a virus is budding at the plasma membrane, that's the way it is all the time. It doesn't change. And the nucleocapsids inside can have helical or icosahedral symmetry. So here in my hand is a icosahedrally built virus. You can have an envelope around it. Or, again, the helically built nucleocapsid could have an envelope around it. So this one on my right hand, the helical one, is only naked in plant viruses. In animal viruses, it's envelope. These icosahedral capsids can be naked or envelope. And this diagram at the bottom just shows you the process of budding wherein a virus acquires an envelope from the plasma membrane. And that's something we'll talk about in more detail later on. Now, these envelopes that are derived from the host cell, they have to have viral proteins in them to allow them to attach to receptors on host cells. If it were just a naked envelope, it would be hard for it to infect a new cell. And these are called integral membrane glycoproteins. They are diagrammed on the right here. Here's a typical lipid bilayer. These are transmembrane glycoproteins. There's an external domain. There's an internal domain. If this were the virus membrane, this would be the interior of the particle. This would be the exterior portion. The internal parts are typically involved in assembly of the virus particles. And as we'll see, the external domains, they're involved in attachment to cells. They're involved in exit from cells. And when you make antibodies against viruses, as you do with vaccines, often the antibodies are directed against these glycoproteins. Now, they are repeated many times on the virus membrane. They're oligomeric, which means they can be composed of multiple subunits, and they're repeated. So if you look at an electron micrograph of these viruses, they form distinct structures. So here's influenza virus in an EM, and you can see there's some kind of striation going on the outside of the membrane. Those are individual spikes made up of trimers of a surface glycoprotein. And we call those spikes because originally they looked like spikes and the name has stuck. So viral envelope glycoproteins we'll talk about quite a bit. These glycoproteins can be oriented perpendicular to the membrane. So influenza virus has a major glycoprotein in its membrane called the hemagglutinin, or HA. You'll see this many times in this course. It is a trimer embedded in the virus membrane. It's the way the virus attaches to host cell receptors, but it's perpendicular to the membrane. And here on the bottom, lower left, you can see a model of influenza virus. The HA molecules are in blue. You can see they're sticking up from the membrane. But not all viruses have this orientation. In some, the glycoprotein is actually parallel to the membrane. So that's shown on the right, panel B. This is the E glycoprotein of flaviviruses, including dengue, West Nile virus, Zika virus. The glycoproteins, it's also an oligomer. It's lying parallel to the to the virus membrane. And in this picture at the bottom here, you can see the glycoproteins laid out in different colors. So very distinct appearances to the particles as a consequence of this. When we look at all the envelope viruses out there, we can put them into two general categories based on what is inside the envelope. So viruses, envelope viruses with helical nucleocapsids. We've talked about a bunch of these influenza virus, Ebola virus, and other filoviruses, measles, mumps virus, etc. They have a helical genome, helical nucleocapsid with a membrane. The glycoproteins in the membrane don't have any particular symmetry when you look at the particle. They're not arranged with icosahedral symmetry. If you look back at the influenza structure here, you can't see any symmetry in the way these glycoproteins are arranged on the surface. But there are also viruses where the envelope is around an icosahedral capsid. And because the membrane glycoproteins can contact the underlying icosahedron, it gives them some symmetry. So here, for example, is an example of such a virus where there is an icosahedral capsid surrounded by a membrane. Within the membrane are glycoproteins, and the glycoproteins assume the symmetry of the underlying capsid, which you can see here in the left-hand picture. It's a bit dark, but there's a 5-fold axis of symmetry. You can count 5 subunits around it. There are also 2-fold and 3-fold axes. So this is a structured envelope because the glycoproteins, they could be floating around in the membrane, but they don't because they're interacting with the capsid underneath. And that is why this HIV structure is wrong. This is an unstructured envelope. It has glycoproteins in it, which do not have any symmetry at all resembling an icosahedron. Yet, the makers of this, you can see, have given it a 5-fold axis of symmetry. Okay? Why is that? Well, it turns out many years ago, someone drew a model of HIV, an artist, and he or she drew it with 5-fold symmetry. And now the makers of this just picked that off the internet and modeled their fuzzy toy based on it. But it's not right, and nobody will fix it no matter how much I complain every year. So we're stuck with that. But now you know, if you give it to your nephew one day, you have to tell him or her why it's wrong, okay? All right, structured and unstructured envelopes. That brings us to these big viruses that are built using principles we haven't discovered yet. We call them complex because we don't understand them. And they include pox viruses, which has multiple membranes and an interesting core, but no symmetry that we can tell of. Pandora viruses, the big ones, micron in length, which don't seem to have any symmetry. They have an apical pore at one end, which is probably involved in the genome getting in and out. And in the intact virus particle, this seems to be plugged. This pore seems to be plugged with some material, which you can see in the electron micrograph on the upper right. And the discoverers of this have, of course, they're French, so they called it a cork. But these don't have any symmetry that we can understand. They look more like cells in some ways in the way that they're built. And finally, there are lots of other things in virus particles. Besides structural components, there are enzymes of different sorts that we'll talk about, polymerases, integrases, proteases, polypolymerase capping enzymes, topoisomerases, all these involved in processing or nucleic acid biosynthesis. There are activators of mRNA synthesis, enzymes that degrade mRNA, things that are needed for infection, and even some cellular components, tRNAs, histones, a variety of lipids, and many more proteins. In fact, one of the interesting things that is being done is to take different viruses, do mass spectrometry on the purified components, find out what's in them, and figure out what they do. So, of course, these other components don't fit these symmetry rules that we've talked about, but they certainly have other roles in virus structure.